biology students to another riveting installment of Advanced Biology lesson. We have been slowly walking through the macromolecules, so we have been taking them one at a time. We have learned about carbohydrates, we've learned about lipids, and today we are going to take the big step and learn about my favorite, if there ever was a favorite macromolecule, this would be it. We are going to talk about proteins. Proteins. I wrote this up here, and this sort of underestimates how cool a protein is. It says proteins are an important molecule in carrying out the functions of a cell. But proteins are way more than an important molecule. Proteins are critical to cell function. Without proteins, cells would cease to exist. They would be able to do nothing. They wouldn't be able to make energy. They wouldn't be able to maintain homeostasis. They wouldn't be able to let things in and out of the cell membrane. They would be defunct. So proteins are critical to making sure a cell works the way it does. For instance, if a cell wants to send a message, it sends it by a protein. If a cell wants to receive a message, it receives a message because a protein receives a message. If it wants to convert glucose into ATP energy, it's going to use a protein to convert glucose to ATP energy. Actually, it's going to use multiple. So proteins are critical in carrying out the functions of a cell. And some important proteins, right? We get our proteins, so we get some of our proteins by eating them. We need to ingest proteins. And down over here, this is a little reminder. My pointer's a little off here. We'll see if this works. Down here's a little reminder of biology for you. That we talked about proteins are made up of these amino acids that are linked together to form large chains. So I sort of want to start with that understanding and build on that today. So let's look at this. So they are formed from the building blocks amino acids. So our monomer for proteins are amino acids. Our monomers are amino acids. Here is the structure of an amino acid. And there are three groups. This group right down here is what we call the amine group. Okay, so there's an amine group. There is over here, this group right here is called our carboxyl group. Right, we're familiar with those C double bonded OOHs, the acid group. Right, amine group, acid group, amino acid. And then there's also this group down here, and this is called the random group. And the random group changes depending upon which amino acid it is. So the other parts of the molecule are always the same. There's always NH2, there's always a central carbon with hydrogen, there's always a carboxyl group. Always. They're always the same. The only thing that changes, whether we're talking about leucine or galline or all these different amino acids, the only thing that actually changes is this R group right here. Okay? So, when we have these amino acids are going to link together to form large chains called polypeptides. And they're actually linked together with little bonds here. These little bonds in between each amino acid. And those are called peptide bonds. And we'll talk about those in a little bit. But these polypeptides, these long chains of amino acids right here, we can see some of them. Those are called a polypeptide. Now, polypeptides then form 4 levels of structure. Got deleted here, but there are 4 levels of structure. The first one, it's called primary structure. So if we look up here, here is our primary protein structure, and that's just a long chain of amino acids. That long chain of amino acids begins to coil up on itself, and that forms this secondary protein structure. So primary is a large chain. Secondary are these coiled chains. Tertiary, these things begin to coil up so much that they begin to bend and fold on each other. And this bending and folding, there's actually bonds that form within the molecule. This bending and folding is called a tertiary structure. Now it looks like a bunched up a bunch of chords that are lumped together, so it forms a clump. This clump meets up with other clumps. So if you notice down here, there's not just one, but there is actually two clumps here, two different clumps, and that's our quaternary structure. So we get multiple of these tertiary polypeptide bunches, and that forms our quaternary protein structure. Okay? Once again, we take a look at this. Primary, long chain of amino acids. Those amino acids begin to coil up to form our secondary. Those coils begin to bunch up to form our tertiary, and those bunches find other bunches to form our quaternary protein structure, or our actual completed functional protein. So this right here is what the proteins in your body look like. They are these quaternary functional proteins that do everything we need them to do. Okay, so we're going to stop there for today, class. If you have any questions, ask me anytime. I hope that was informative. I look forward to hearing what the Protein Master Group came up with, and that's exciting for us. So have a good day, team. We'll see you soon.